Hello, welcome everybody. Thank you for joining us today for this webinar. My name is Jim George and I'm the Market Development Manager for our biopharma group at Thermo Fisher. It is my pleasure to be hosting our webinar today, introducing the HLA assay as a companion diagnostic in immunotherapy development. So I'd like to introduce our two panelists today. Leanne Zanetti is the Senior Director of Global Lead Precision Medicine at Novartis. She has worked in the field of molecular diagnostics for 18 years with a focus on oncology and companion diagnostics. Her role is focused on driving the uptake of quality diagnostic testing so that all patients eligible will have access to the right drug at the right time. With the varying levels of adaptation and reimbursement of biomarker testing globally, ensuring global testing access means working across multiple technology platforms, including next-generation sequencing, IHC, qPCR, and liquid biopsy. Leanne is also a board member at From Testing to Targeted Treatment, a multi-stakeholder collaborative program that aims to unify, accelerate, and build on existing efforts to make precision medicine a reality for all patients who could benefit from it. It's co-led by patients. FT3 is strongly focused on patient needs and experience. Our other panelist is Sean Carey. He's the Scientific Affairs Senior Manager for Transplant Diagnostics section of Thermo Fisher. Before taking this post in May of 2022, Sean was a consultant clinical scientist responsible for the histocompatibility and immunogenetics laboratory at the University Hospital Plymouth in the United Kingdom. Sean's career in the transplantation laboratory began at Guy's Hospital in London in 1992, where he was employed to investigate the efficacy of immunosuppressant drugs and the immunological desensitization of highly sensitized renal patients. Sean moved into the routine transplant laboratory at Guy's in 1996 and completed his master's in immunology the following year. Taking the role of chief biomedical scientist at the University Hospital in Plymouth in 2022, Sean implemented flow cytometry cross-matching and led the team to introduce many of the One Lambda Suite Luminex testing products. Following a few years in senior laboratory management, Sean focused his attention back on transplantation, and in 2014, as lead healthcare scientist in H&I at the University Hospital in Plymouth, commencing a doctorate course with the University of Manchester at the same time. He achieved Doctor of Clinical Scientist in 2020 and was admitted as a Fellow of the Royal College of Pathologists the same year, having completed the examinations during his doctorate. Sean has published extensively throughout his career, beginning with first author paper in 1992. His doctorate research led to 3 first author publications, including a review of HLA expression in 2019, which has been cited almost 50 times to date. It is my pleasure today to turn the presentation over to Sean Carey.
Thank you very much. I'll just share my screen. Hopefully that everybody can see that. Thank you very much for that introduction. And I'm very aware that our audience today is quite varied. So I'm going to try and give an overview of HLA for those new to HLA, and then an overview of where that interacts with both biopharma, but also some of the other industries that use HLA as its help to try and understand what's going on either in disease association or in transplantation, so that we really get a feel for why the HLA system in humans is so central to many of the diseases that we go through, but also the treatments for the diseases as we go on from there. So basically, I'll just introduce HLA to the whole audience and explain the different classes, class 1 and class 2, and then just give a little overview of why HLA is important in transplantation, how it's relevant in disease associations and some drug interactions, and then just summarize in how it's being used nowadays in biopharma, and then I'll introduce Leanne, and we can then open up the discussion from there. So HLA is coded on chromosome 6 of the human chromosome. And the important thing about that is firstly that it is codominantly expressed. So both copies of the chromosome are translated, and both of the sets of genes are expressed on the cell surface. HLA is comprised of two regions: HLA class 1, which is made up of three loci, HLA-A, B, and C, and class 2, which is made up of also three loci, HLA-DR, DQ, and DP. There are slightly more loci within each of the HLA areas for each of the DR, DQ, and DP, and I'll explain what that is as we go through. So starting with HLA class 1, so that's HLA A, B, and C. The coding for the molecules is based on obviously a series of exons and introns. But the interesting thing with HLA, both class 1 and class 2, is the first exon is actually cleaved off. It is used elsewhere in part of the immune machinery, but for the sake of this talk, basically, we're interested in exons 2 right through to exon 8 for class 1. The final protein is made up of a shape that forms basically a binding cleft made out of exons 2 and exons 3, and this is bordered by two alpha helices and a beta pleated sheet in the bottom. You can see it more closely in this diagram here. And then the HLA molecule actually presents some peptide from the cell actually in that cleft. The whole protein is held structurally via beta-2 microglobulin, and then there is some signaling that goes through the cell membrane. HLA class 1 molecules are found on almost every cell in the body, and they have a role basically to interrogate the cell contents of the cell to make sure that it is not pathogenic in any way. So it could be have been overtaken by viral machinery to make more viruses, or it could be perhaps mutated and form some form of cancer going on inside the cell. As the cell is walled, then there is not normally any way to interrogate the inside of the cell, and that's the role of the HLA class 1 molecule. What it does, it breaks up peptides or proteins in random into short peptide sequences of between 12 and 20 amino acids long, can be longer. And these peptides are then loaded onto that groove of the HLA class 1 molecule, which works its way and is then presented on the surface of the cell. If we remember, there were two HLA-A, two HLA-B, and two HLA-C for class 1. So potentially there are 6 different molecules on the cell surface, all presenting peptides from inside the cell. HLA class 2 is similar. Again, it's made up of obviously of exons and introns in terms of the DNA. And again, exon 1 is lost. But in the case of class 2, it's not a single molecule held together with beta-2 microglobulin, but it's actually 2 molecules, the alpha chain and the beta chain, that are bound together in a similar conformation to HLA class 1, in that they have the alpha helices and the beta pleated sheet that forms a binding cleft again, but this time this structure is formed from the 2 exons 2 of the alpha chain and the beta chain. They have a slightly different role in our immune system, in that they're not looking inside the cell, but they're looking externally at into the blood, in the lymph, in the extracellular spaces. And they work by basically by pinocytosis. They absorb potentially foreign antigens, very often by binding to antibody. This is absorbed inside the cell. The proteins are then broken up inside these bodies, and then again go into the Golgi apparatus. And the peptides that are made from digesting that protein that's been brought from outside the cell is then loaded onto the class 2 molecule. And again, that's presented on the cell surface in a very similar way to class 1. The key to difference between class 1 and class 2 is obviously this cell itself is perfectly intact, and it therefore has no issue with it. It's not got a virus in it or a pathogen in it. But actually what's being presented are peptides from the immune system, from outside the cell. This cell, of course, will have HLA class 1 molecules on it as well. So therefore, it's continually being interrogated internally to make sure that it's fine as well. So in terms of what the two molecules do, HLA class 1 consists of 3 groups, A, B, and C, and is designed to present intracellular peptides as part of the immune surveillance. HLA class 2 is designed to present extracellular peptides, and that's in, again, 3 different flavors, HLA DR, DQ, and DP. And the way they work is by these two. I'm sorry, this picture is just a little bit busy, but basically the HLA class 1 molecules, T cells in the form of cytotoxic or CD8 T cells, have been educated in the thymus to recognize self-peptides and therefore not respond to self-peptide. But therefore, when they interrogate the HLA class 1 molecule here, if there are viruses or pathogens inside the cell that are presented in the form of peptides on the HLA molecule, then the CD8 cytotoxic T cell will recognize those, especially in the presence of other inflammatory cytokines, and that will lead on to killing of the virus-infected cell. Similarly, antibody production, so antibodies that have been produced that recognize viral or other pathogens in the extracellular spaces, they will absorb those pathogens into the antigen-presenting cell, and again, that peptide from the pathogen is presented, but this time to a helper T cell, CD4 T cell, and what that does is it stimulates the antigen-presenting cell to go on and produce more of itself, and therefore lots more antibody to get into the extracellular spaces to then go and find more of those pathogens. So HLA class 1 results in death of the cell. HLA class 2 leads to antibody-mediated immune responses. But in both of those cases, the HLA molecule is absolutely central for recognition of those foreign peptides. So this is an example of different HLA molecules, or the DNA for them. Exon 1 is here in pink. The important thing that you see is that actually the molecule largely consists of a backbone of exons in class 1, exons 4 onwards, that is largely invariant, although it does vary occasionally. But it's exons 2 and exons 3 that are the most variable of the in terms of the DNA, and therefore the shape of the final protein is the most variable in those exons as well. And if you remember, those are the parts of the protein that hold on to the peptide. And you can see that the more and more that we use sequencing to look into populations across the world, the more we find that there are different HLA proteins, and therefore those peptides, mainly in exons 2 and 3, those peptides are being found across the world in many different forms. And the reason for that is that the immune system is continually trying to evolve to deal with the pathogens that are present in the local environment. And so we have a space-filling model here of the looking down on the top of an HLA class 1 molecule. And you'll see in gray here, the peptide is sort of held inside that molecule. And the way that the peptide is anchored into the HLA molecule is usually for HLA class 1 by the, in terms of the peptide, both the second and the last position of the peptide. So they act as anchors for that peptide within this HLA molecule, and the rest of the peptide can actually be quite variable. And the importance of that is that as our HLA system is evolving, we're trying to present different peptides. The world around us in terms of pathogens, and we just had the COVID-19 outbreak, the world around us, the pathogens are also evolving. And so our immune system over many generations and very slowly, but continually, is trying to present local pathogens as well as it can to our immune system so that our immune system can deal with those quickly. And therefore our genes will survive and be passed on to our families. And so across the world, we're finding many different HLA molecules able to present different peptides to our immune system, and therefore, as a world population, in effect, we're able to deal with all the pathogens that the world is trying to throw at us. So why is this important? Why is it relevant? And I'll just go through the different cases now. In medicine, firstly, in transplantation, the biggest difference between cells of a transplant organ and the recipient is the HLA molecule. That's the thing that varies most between individuals. And as I showed you earlier, there are many thousands of different HLA molecules. Our T cells are educated in our thymus to recognize the peptides presented on our HLA molecules. And the way they do that is as they go through the thymus, they have to be selected to be able to contact our HLA molecules and then see whether the peptide that those HLA molecules is presenting belongs to us, in which case the T cell's happy and goes elsewhere, or doesn't belong to us, in which case immune response results. The difficulty in transplantation is that the antigen-presenting cell, especially to start with, is from the donor. And if the HLA is not exactly the same as the recipient, then the binding of the T cell receptor to that donor HLA can be too strong and therefore create a positive signal, even in the absence of any signal being created by the peptide itself. So it's actually the HLA T cell receptor interaction that's going wrong in that case. And that very often can lead to T cell mediated rejection. Later on in the transplant process, the donor antigen presenting cells are replaced by recipient antigen presenting cells. These can then load donor-derived peptides in the same way as you would if they were an invading pathogen, in a sense. That's then presented onto the T cell receptor in the same way as a normal response. It's recipient HLA, it's recipient T cell receptor, and that then goes on to lead to quite often an antibody-mediated response. And as we see in transplantation, the more mismatches, so MM stands for mismatch, the more mismatches there are between the donor HLA and the recipient HLA, then the worse the transplant outcome. So our job in the transplantation laboratory is trying to match the two as best we possibly could. HLA has a relevance in disease association, and many of these diseases are probably the result of a slight autoimmune reaction or the immune system being overreactive in certain individuals. And I'll go through a few of these, but there are quite a lot of disease associations, some very common, some rarer, and they require very, very specific HLA molecules for those diseases to be manifest. Celiac disease is very common in the West nowadays. So this is how we sell celiac in the UK. So I do apologise to those of you that are not from the UK. The normal villus of your intestines should have a high surface area to allow absorption of all the foodstuffs that we eat. But in celiac disease, this is sloughed off and destroyed, and the surface villi are seriously damaged, and that leads to digestive problems for the sufferers. And what we understand is happening in this is that tissue transglutaminase deaminates the normal gliadin peptides into a form that we've not been, or our immune system doesn't recognize. The peptides that have been deaminated bind strongly to HLA that are associated, and this causes an autoimmune reaction and autoantibodies. And it needs a correct DQ alpha and DQ beta genes together So DQ alpha 105 with the DQ beta 102 together forming that peptide, and that holds on to that particular peptide in an unusual way to our immune system that creates this immune response and local damage, or an alpha 103 with a DQ beta 10302. So they're very unique combinations, and they need to be together for that disease to occur. Ankylosing spondylitis is also quite a common disease. It causes back pain in early presentation, but actually leads to quite severe deformation of the sacroiliac joints later on in life. And it takes many, many years to manifest. We think with this disease, normal pathogen presentation of maybe an infection early in life on an HLA-B27 molecule leads to some sort of autoimmunity. Maybe the peptide looks similar to one of our own normal peptides, and this potentially leads on to an autoimmune disease where we start reacting to self rather than to anything foreign, and that reaction seems to be ongoing and uncontrollable, and over many years it actually leads to a self-destruction in a way by our own immune system. And the final one I'll talk about is Stevens-Johnson syndrome, or severe cutaneous adverse reactions. And there are quite a few of these. And as we are using more and more drugs, we're finding more and more drug HLA interactions that cause this disease. It needs the presence of the drug in the body with a very unique HLA type to cause it. HLA-B5701 with abacavir, which is an HIV drug, is one of the earliest ones to be recognized, but there are other ones that have also been recognized. Treatment is to stop the drug, and that stops the symptoms. And the reason for that is that the HLA molecule, you can see here, is presenting a peptide normally, but the drug gets in underneath the peptide, we understand, and that pushes the peptide into a slightly different conformation. The T cell receptor therefore doesn't recognize the peptide as being self anymore, and reacts to this drug peptide interaction or combination as foreign, therefore causes this massive reaction. And finally, HLA in biopharma. So tebentafusp is a drug that is being designed to treat uveal melanoma. Now, uveal melanoma is a rare disease. It's got an incidence of about 5 per million in the US, but it is the primary cause of primary ocular malignancies. Glycoprotein GP100 is a normal protein in the body, but it's highly expressed in these particular melanoma cells. So tebentafusp is designed, it's a monoclonal T cell receptor that recognizes the GP100 peptide, but only in the presence of HLA-A201. So it's that unique combination of the HLA molecule with the peptide inside it that T cell receptor recognizes. And then the other end of the drug has a T cell receptor on it to bring the T cell close to the cancer cell, and therefore the T cell can then go on and enact its normal killing methods, usually through cytokines and chemokines. So this is what it looks like. You have the T cell receptor end here that recognizes the GP100 peptide in the presence of the HLA-A201 molecule. And then on the other end, a T cell receptor that brings the T cell close to that cell, that enables the T cell to enact killing of the cancer cell. HLA-A*0201 is present in about 50% of most populations, and therefore this combination was selected because it's able to treat such a high number of people across the world. Unfortunately, it doesn't obviously work with anybody that doesn't have HLA-A201, so it's important to do that HLA typing up front before the drug is given. But in trials, it doubles the number of people that go into either a complete response or a partial response compared to a control group of individuals. And there are some treatment-related adverse events because the GP100 is normally present in the body, and therefore that peptide is presented on other cells, but in much lesser amounts than on the malignant cells. And I'll finish with this final example of chimeric antigen receptor, or CAR T cells. And that's presented in a similar way. In this specific example, that's still undergoing trials at the moment, there is a very specific mutation in protein in many cancers. So the wild type peptide, you can see I've just highlighted the position 2 and the final position anchor points here. But the wild type, which has an R in this position, is mutated to a Q, and but both of those are presented on the HLA-B0702. But the company have used a phage stray library to create antibodies that only recognize the mutant peptide, sorry, this one, presented on HLA-B0702 molecule. Again, this class 1 molecule is very common in many populations, which is why it's been selected. The selected antibody does not recognize the wild-type peptide in the presence of the same HLA molecule. So it's unique to the mutation, and therefore very specific to cells, cancer cells that are presenting this particular peptide. And what it does there, in the same sort of way, it recognizes the tumor antigen through the tumor peptide. It brings the CAR T cell close to the tumor cell that then can enact T cell killing. And these are ongoing clinical trials at the moment. I'm going to repeat this slide because this is quite important. When we go on in terms of working with biopharma, the important thing is obviously to develop the drugs and to have the drugs that can help treat these cancers. But they, in these examples, they are peptides that are presented in combination with very, very specific HLA types. And HLA typing in terms of different populations, whether interested in disease association, whether interested in transplantation, whether interested in these cases in biopharma, the level of HLA typing is really, really important between those different cases. And where we're looking at specific HLA molecules, for example, the A star of 201 compared to in biopharma, in tebentafusp, then we actually need to be very, very unique alpha chain, exons 2 and exons 3. And I've listed in this example different HLA-A2 types, but actually showing you in an exons 2 here and exon 3 here, the different amino acids that actually make up those 2 exons just by the second field of HLA typing. So you can't use any form of HLA typing to do the initial typing of potential recipients. You have to be able to type to the second field, high resolution typing, so that you know that you're getting the right peptide and the right HLA molecule on the cell surface for the drug to be able to react with. And obviously at 1 lambda, we have the fastplex, which gives you 11 loci typing, which gives you all the typing you need for any potential drug. But also, we've just been granted FDA approval for HLA typing using our CCOR CDX typing, which is just a single locus. So that just looks for the HLA A201 DNA type in the potential recipients, and therefore this process, the CCOR CDX, has been granted the de novo classification because it gives us the level of typing that we need to be able to refer patients for uveal melanoma in these cases. So I'm going to finish there and stop sharing my screen and introduce you to Leanne.
Hello, Sean.
Hi, welcome, and thank you very much for joining us. So maybe you'd like to just start by telling us a little bit about your background.
Sure. For those dialing in, they can probably notice that I also have a Commonwealth accent similar to Sean. So I'm from Australia. Following university, I started my career working as a medical scientist in immunology, performing HLA typing actually in a transplant lab. Following that, I progressed into bioinformatics for Sanger sequencing analysis, and then later to next generation sequencing. And following my stint in the lab, I transitioned over to industry, working across Asia Pacific, and then relocated to the US, which is where my global companion diagnostic development and commercialization experience really began. And in my previous role at a diagnostic company, I worked on multiple companion diagnostic programs with multiple pharma partners. So I guess you could say that I've seen the good and the bad. And then I've been able to apply these lessons that I learned across those programs to my current role at Novartis.
Fantastic. Thank you very much. So how do you go developing a companion diagnostic? What are the main considerations during the development?
Sure. So when you're thinking about a successful development pathway for a companion diagnostic, it really relies on a strong collaboration between pharma and the diagnostic partner. In pharma, we need to realize that we are the ones who need the diagnostic development and commercialization to be successful. It's not the diagnostic company. And so therefore, because we're the ones that are really trying to enable access to patients. So therefore, we need to be the driving force behind the availability of the CDX at time of launch. And so therefore, I would encourage, you start with the end in mind. And that sounds quite ominous. What do we mean by starting with the end in mind? You need to really think about where you want to launch this therapy, what technology is required to test for that biomarker, and the list goes on. And from there, you would need to then map out the various regulatory and reimbursement needs across those countries that you had identified. And so basically, I need to think about the market environment that needs to be in place at time of launch to de-risk any of the barriers to testing, at launch and beyond. But you can't do that alone. So when selecting a partner, I consider, some key elements, which are global footprint and expertise. So yes, there is many companies that pop up that have got these really cool technologies. However, they're usually isolated to the single area that the startup is located in. So really looking for a global footprint and expertise, proven capabilities in bringing in IVD to market. I experience across FDA, PMDA, and all the other regulatory bodies. Market access or reimbursement experience, and in an ideal world, multiple modalities. So what I mean by that is it really depends on the biomarker and the patient population. But in some cases, it's helpful to have a lower throughput or simple and higher throughput or complex solution. So in terms of oncology here in the US, I'm actually in Milan, so I'm not really here in the US right now. Community hospitals are where the majority of the patients are located. And the higher complexity testing is centralized at academic centers. So the testing is, in general, not really close to the patient. So in an ideal world, you would have a diagnostic partner that can offer these two modalities. That's my preference.
Okay, thank you very much. And in terms of regulatory issues, we in the H&I world, thought that we had it all sort of fairly sewn up because we knew what we were using it for, and we were, everything's all about HLA, and now we're learning again how to work with partners in other fields to actually create something that spans both of the areas. In your sort of experience, how difficult is that to bring those two things together?
I think I'm getting what you mean by this. So in terms of a regulatory hurdle, considering my background from HLA and what I know currently in the pharma field, I think you're, correct me if I'm wrong, I'm going to go down one track, but we can always circle back. So it's more the nature of developing a biomarker-driven therapy, and sometimes it's not 100% certain if the diagnostic is required. and that's usually determined at a very late stage by the regulatory authorities. So the challenge is with that regulatory hurdle looming is, again, start with the end in mind. So de-risking your program, and that really prevents a fire drill or post-marketing commitments, which can actually drive up costs.
Yeah.
Because of course it is 100% possible to accelerate a CDX in a short time period, but that will then require more staffing and resourcing on the back end from a diagnostic provider. And so the savings that are attributed to not investing as early and waiting for the regulatory feedback is really a false saving, because anything that you accelerate has a cost at the end of it. So I don't know if that's what you're getting at, but that is something that I would see as a major challenge from a HLA CDX.
That's great. Thank you very much.
Awesome. Well, thank you very much. Thank you very much, Leanne and Sean, for this wonderful webinar. I'd also like to thank all of our attendees. Hopefully you found this very engaging and informative. Please keep a lookout. There should be a survey that will be sent out to you to help us improve events for the future. And with that, I'm going to, again, thank Sean and Leanne, both of you over in Europe. Enjoy the rest of your evening. But thank you very much for taking the time to help us out with this. And with this, I'll conclude the webinar and wish everybody a wonderful afternoon, evening, morning, wherever you're at. Thank you.
Thanks so much, Jim.
Thanks so very much.
Sean.